So SOLTI VALENTINE is a neoadjuvant study testing HER3-DXd, an antibody drug conjugate directed against HER3 and with a payload of DXT in patients with high-risk HR-positive, HER2-negative early breast cancer. So the objective of SOLTI VALENTINE, which builds in previous results from another trial from SOLTI HER3, is to assess the efficacy of this potent anti-HER3 ADC in patients with high-risk disease, which are a population of patients in need for better treatment strategies. So this is a randomized phase 2 study with 3 arms, one arm with a HER3-DXd a single agent, another arm of HER3-DXd in combination with letrozole, and a third arm with standard multi-agent chemotherapy based in anthracyclines and taxanes. Patients had tumor collected at baseline, at cycle 2, day 1, and at surgery. The primary endpoint of the trial was the PCR rate in each of the arms. And we also looked at several efficacy endpoints like overall response rates. We will also look at invasive disease-free survival, also safety endpoints and translational endpoints like reduction in Ki-67, change in the PAM50 subtype, and change in the risk of recurrence score by PAM50. So in SOLTI VALENTINE, we observed PCR rates and an overall response rate with HER3-DXT, either alone or in combination with letrozole, that was similar to patients treated with multi-agent chemotherapy. Additionally, it was also very interesting to see that the proportion of patients having grade 3 or higher adverse events with HER3-DXd was substantially lower than the proportion of patients receiving multi-agent chemotherapy. There were also less treatment discontinuations and dose reductions in those patients treated with HER3-DXd. The PCR rate was low overall, but I think it reflects the biology of the tumors enrolled in VALENTINE, and it was very similar with multi-agent chemotherapy. The other interesting part of SOLTI VALENTINE is the translational research that we have already presented and is currently also work in progress. But we have observed a very nice reduction in Ki-67, translating good anti-proliferative response of HER3-DXd, both alone or in combination with letrozole. We also saw change in PAM50 subtype from a more proliferative luminal B to less proliferative luminal A and normal-like subtypes at surgery, and also a decrease in the risk of recurrence scores, and a correlation of the cell TIL score, which is a composite score of tumor TIL infiltration and cellular composition in cycle 2, day 1, with overall response rates in the patients treated with HER3-DXd, and this confirms prior results obtained from the HER3 trial with just 1 dose of HER3-DXd. So I think that SOLTI VALENTINE suggests that HER3-DXT may be treatment options for patients with high-risk HR-positive, HER2-negative early breast cancer. And I believe that these results support the continuation of the development of the drug in this context, perhaps as a part of a neoadjuvant regimen that has at least the same activity but is better tolerated for this high-risk population.